Hello, Dr. Hamid is here. Today we are going to talk about cholinergic receptors. Cholinergic receptors are a type of receptors which respond to acetylcholine. So cholinergic receptors respond to acetylcholine. It means that acetylcholine can bind to different type of cholinergic receptor, which today we are going to talk about it. And it can activate the signaling pathway inside or signaling molecule inside the cell, and it can show its effect on the cellular level. Now, before talking about the cholinergic receptor, first we should know about what is acetylcholine. Acetylcholine is a neurotransmitter in the cholinergic fibers. It means that cholinergic fiber can make acetylcholine and can release acetylcholine in the synapses, and by releasing acetylcholine they can affect other neurons or target cells. So I said that acetylcholine is the neurotransmitter in cholinergic fiber. Which fiber can make acetylcholine and can release acetylcholine? Cholinergic fibers. First is all paraganglionic fibers can make acetylcholine and can release acetylcholine. What is paraganglionic fiber? If you know, we have ganglion, which is a collection of cell body of the neurons. The fibers which are before this ganglion, for example, imagine that this fiber is from sympathetic nervous system, or this fiber is from parasympathetic nervous system. It originates from its origin, then it synapses, it makes synapse with the second order neuron, which is after this ganglion. The fiber which is before this ganglion, this fiber is paraganglionic fiber. Paraganglionic fiber. So what are paraganglionic fibers? Paraganglionic fibers are the first order neuron which originate from its origin and synapse with the second order neuron in the ganglion. These are the paraganglionic fibers. No matter if it's from the sympathetic nervous system or it's from the parasympathetic nervous system, all paraganglionic fibers, they can release acetylcholine as a neurotransmitter. What is postganglionic fiber? Postganglionic fiber are the fibers which are after the ganglion. These originate from the ganglion and ends to the target organ. For example, now here target organ is the eye. These are the postganglionic fibers. Postganglionic fibers. Now, the first group of cholinergic fibers were all paraganglionic fibers. The second group of cholinergic fibers are postganglionic fibers of parasympathetic nervous system. Postganglionic fibers of parasympathetic nervous system. Parasympathetic nervous system. Imagine that this postganglionic fiber is from parasympathetic nervous system. When it ends to the eye, it can release acetylcholine as neurotransmitter, and this acetylcholine binds to different type of cholinergic receptor and it can affect the target organ. Here also we release acetylcholine in the ganglion. So the first group of cholinergic fibers were all paraganglionic fiber. The second was postganglionic fiber of parasympathetic nervous system. The third group is postganglionic fiber of sympathetic nervous system, which innervates the sweat gland. Postganglionic of sympathetic, which innervate sweat gland. postganglionic fibers of sympathetic to sweat gland. These are also cholinergic fibers which release acetylcholine as the neurotransmitter. Imagine that these are the fibers of sympathetic nervous system, and these fibers innervate sweat gland. These fibers also release acetylcholine as neurotransmitter. And after releasing acetylcholine, this acetylcholine binds to the cholinergic receptors on the sweat gland, and it can increase sweating. So the third group are these type of fiber. And the last group of cholinergic fibers are somatic fibers. Somatic fibers are a group of fiber which innervate the muscles. Imagine that this is the muscle. This is the muscle. These somatic fibers directly innervate the muscles and it can stimulate the muscles, it can affect the muscle by releasing acetylcholine as a neurotransmitter. Then this acetylcholine binds to the nicotinic receptors in the neuromuscular junction, and it can stimulate nicotinic receptor there. So these were cholinergic fibers. The first group is all preganglionic fiber. The second, postganglionic fiber of parasympathetic nervous system. The third, postganglionic of sympathetic nervous system, which innervate sweat gland. And fourth is somatic fibers, which innervate skeletal muscles. Now we know which type of fiber are cholinergic fiber, and they release acetylcholine as a neurotransmitter. The second thing we should know about cholinergic receptors are a type of cholinergic receptor. We can divide cholinergic receptor into two types. One is nicotinic receptors, and second is this type of receptor which are muscarinic receptors. And these two type of cholinergic receptors are named after nicotine and muscarine alkaloid. If it can bind to the nicotine and it can respond to the nicotine, those are nicotinic receptor, and if it can bind to muscarine and respond to muscarine, those are muscarinic receptor. First, we'll talk about the nicotinic receptor. This is a nicotinic receptor which is made of 5 subunits. 2 subunits are alpha subunits, one is beta, the fourth is delta, and fifth is gamma. So these 5 subunits are arranged around a central core, and they can make a channel. This channel in normal situation is closed, but when acetylcholine binds to its site or its location, the location or site of binding acetylcholine is between the alpha and gamma subunit and alpha and delta subunit. When acetylcholine binds to these two sites, it can open the channel. By opening the channel, we can see flowing down of sodium. So when I open this channel, imagine that this is the channel. When I open the channel, and this channel is sodium channel. When I open the channel, sodium will flow down, and this is the membrane. Sodium will flow down, and positive charge inside the cell will increases, and this positive charge can cause depolarization. So when acetylcholine binds to its site, it can open the channel. By opening the channel, we can see flowing down of sodium, and this sodium can cause depolarization of the cell. This was about nicotinic receptor. But based on the existence of agonists and antagonists, which can distinguish the nicotinic receptor, we have 3 subtypes of nicotinic receptor. One subtype is NN, which this N is for neuronal. It means that we can find this subtype in the ganglion as well as in the adrenal medulla. So NN subtype is in the ganglion and adrenal medulla. The second subtype of nicotinic receptor is NM subtype, which this M is for muscle or muscular, which we can find this subtype of nicotinic receptor in a neuromuscular junction. And the last is a specialized or a special subtype of nicotinic receptor which is located on the brain. Why am I saying a special subtype? Because this subtype is a perisynaptic receptor. Perisynaptically receptor. What does it mean, perisynaptical receptor? I will draw it here only. We know that neuron can connect with each other through synapses, and synapse is the gap between two neurons. Imagine that this is the axon, and it can be dendrite, it can be axonal membrane of adjacent or neighboring neuron. This is the second neuron. This neuron, which is before the synapse or synapse, this is perisynaptic neuron. And this neuron, which is after synapse, this is the postsynaptic neuron. Most of the time, these nicotinic and muscarinic receptors are a type of postsynaptic receptors. But this subtype, which I told you it is a special subtype of nicotinic receptor, is a type of perisynaptical receptors. It means that when we release acetylcholine in the synapses, this acetylcholine will bind, it can bind to this subtype also, it can bind to post-synaptical receptors also. But these perisynaptical receptor, when acetylcholine binds to this receptor, it can increase releasing of glutamate and dopamine. So, binding of acetylcholine to the nicotinic subtype, which is located in the brain, it can mediate increasing of releasing of glutamate and dopamine. Because of that, when we smoke, the nicotine which is in the smoke, it can bind to this subtype, and by binding to this subtype, it can increase glutamate and dopamine level of this neurotransmitter in the brain, and we can see the effect of this neurotransmitter also. So this was about nicotinic receptor subtypes. We have three subtypes, NN, we have NM, and also we have another subtype which is located in the brain. Next thing we should know about the nicotinic receptor is about location. Just right now I told you about the location of nicotinic receptor. They are in neuromuscular junction. We can find them in the ganglion. I explained this. We release acetylcholine in the ganglion. The type of receptor, the type of cholinergic receptor which is in the ganglion are nicotinic receptor. We can find them in the adrenal medulla also, and we can find them also in the brain, which they are located presynaptically, and they can mediate releasing of glutamate and dopamine. The last things which we should know about nicotinic receptor are about agonistic and antagonistic of each subtype of nicotinic receptor. Told you that we have three subtypes, NN, NM, and the subtype which is located in the brain. First, we'll talk about the agonist and antagonist of NN subtype. I write agonist by red color and antagonist by green color. The agonists for NN subtypes are DMPP. What is DMPP? Dimethylphenylpiperazinium. Dimethylphenylpiperazinium. Phenylpiperazinium is the agonist for NN receptor, and by it can stimulate or it can activate the NN receptor, and it can show excitatory effect on the cell. But the antagonist for NN receptors, they can bind to nicotinic receptor, the NN subtype, but they can not show any intrinsic activity. These are hexametonium and trimethaphone. These two drugs are ganglion blockers. Do you remember I told you that NN subtype is located on the ganglion? These two drugs, they can block the NN subtype, and we call them ganglion blockers. The second subtype was NM subtype. The agonist for NM subtypes are PTMA. PTMA is abbreviation for phenyltrimethylammonium. Phenyltrimethylammonium. This is the agonist for NM subtype. An antagonist for NM subtype are alpha bungarotoxin, and D-tubocorarin. Maybe you know about D-tubocorarin. D-tubocorarin is a neuromuscular blocker. NM receptor is located in neuromuscular junction. And this D-tubocorarin, it can bind to NM subtype, it can block the NM subtype, and it can cause paralysis or it can cause relaxation of skeletal muscle. It depends on dose of D-tubocorarin which we use or which we give to the patient. So D-tubocorarin is a neuromuscular blocker. It is antagonist for NM subtype. Now we'll talk about the second type of cholinergic receptor, which are muscarinic receptor. You can see the difference between nicotinic receptor and muscarinic receptor. Nicotinic receptors are ion-gated channel receptor. But nicotinic receptors, as you can see, these are a type of G protein coupled receptor. G protein coupled receptors. It means that in the intracellular domain, they are coupled with G protein. G proteins are four in types: GS protein, GI protein, GQ protein, and G0 protein, which we will talk about some type of this G protein and the type of signaling molecule which they can activate. Okay, as I said, muscarinic receptors are G protein coupled receptor, which generally we have these receptors on the gland as well as on the smooth muscle. And again, on the basis of the agonist and antagonist drugs, which they can distinguish the different type of muscarinic receptor, we can divide muscarinic receptor into 5 subtypes. One is M1, the second is M2, M3, M4, and M5. These are the 5 subtypes of muscarinic receptors. Okay, now we'll talk about the characteristic of each muscarinic subtype. M1, another name for M1 receptor is neuronal and gastric receptor, because we have this subtype on the neurons in the central nervous system as well as in the stomach. Another name for M2 subtype is cardiac subtype, because we have this subtype of receptor on the heart. Another name for M3 is glandular subtype because we have this subtype on the glands, and we have M4 and M5 in central nervous system. We will talk about this M4 and M5 and the characteristic of these two subtypes. First, we'll start by M1, M3 and M5. M1, M3 and M5, let me write it here only. M1, M3 and M5. These are coupled with GQ protein. And this Q protein, after activation, after binding of acetylcholine to M1, M3 and M5, they can activate GQ protein, and by activation of GQ protein, they can show excitatory effect. Excitatory effect. Now, what will happen after activation of GQ protein? Imagine that this is GQ protein. This GQ protein can activate the enzyme phospholipase C. Phospholipase C. Then this phospholipase C, it breaks down the phospholipid which are in the cell membrane, and by breaking down the phospholipid, it can convert phospholipid. So this phospholipase C, it can convert phospholipid. It acts here only. It can convert phospholipid into diacylglycerol and inositol triphosphate or IP3. It can convert phospholipid into diacylglycerol and inositol triphosphate. Now, what is the action of these diacylglycerol? This diacylglycerol, it can activate another enzyme, that is protein kinase C, and as you know, all kinase enzymes are the type of phosphorylating enzyme. They can phosphorylate different enzyme inside the cell, and by phosphorylating different enzyme, they can show excitatory effect on the cellular level. And what about this IP3? This IP3, it can increase releasing of calcium from sarcoplasmic reticulum. It can increase the calcium level in the cytoplasm, and this calcium, it can activate the enzymes in the cells, in the cytoplasm, or this calcium, it can increase secretion. As you know that we have M1 subtype in the stomach, so it can increase HCl secretion. We have it in the gland, so it can increase secretion and releasing of different hormones. This was about GQ protein. When acetylcholine binds to M1, M3, M5, it can activate GQ protein. GQ protein activate phospholipase C. This enzyme further can convert phospholipid to diacylglycerol and inositol triphosphate. Diacylglycerol further activate protein kinase C, and protein kinase C, it can phosphorylate different enzyme, and it can show excitatory effect on the cellular level. And this IP3 can increase releasing of calcium from sarcoplasmic reticulum. Calcium can increase secretion, calcium can increase contraction, calcium can activate different enzyme inside the cell, and again we can see excitatory effect. But these M2 and M4, these two subtypes are coupled with GI protein. GI is for inhibitory. When acetylcholine binds to M2 and M4 subtype, it can activate the GI protein. Now imagine we have activated the GI protein. When we activate GI protein, this GI, it can inhibit the adenylate cyclase. Adenylate cyclase. And when we inhibit adenylate cyclase, actually we have inhibited conversion of ATP to CAMP. ATP to CAMP. This adenylate cyclase is responsible for conversion of ADP to CAMP, and this CAMP is the second messenger inside the cell. It can phosphorylate different enzyme and it can show excitatory effect. But when we inhibit adenylate cyclase by activation of GI protein, conversion of ATP to CAMP, it won't occur, and the level of CAMP inside the cell will decreases, and that will show inhibitory effect. So M2 and M5, they show inhibitory effect. Inhibitory effect. This was about the effect of activation of these 5 subtype of muscarinic receptor, and also the signaling molecule and signaling pathway which we activate by activation of these 5 subtype of muscarinic receptor. How you can remember where these subtypes are located? What is the most important organ in our body? The most important organ, the first most important is brain. So M1 is located in the brain. The second most important is heart. So M2 is located on the cardiac cells. The third most important organ are the glands. After brain and after heart, glands are the most important. We have M3 on the smooth muscles also. Smooth muscles also. So we can, by activation of these M3 receptor or by stimulating of these M3 receptor, we can see bronchodilation effect, or we can see vasodilatation effect. Also, these are the effect after stimulating the M3 subtype. And M4 and M5, as I told you, are located in the CNS. But something is there about M4 and M5. We don't know about the effect of these two subtypes when we activate them, when we stimulate them. The exact effect of these two subtypes is unknown. Okay, the last thing which we should know about M1, M2, M3, M4, and M5 are about agonistic and antagonistic of each receptor. Again, I will write agonist drugs or agonist chemical by red and antagonist chemicals by green. The agonist for this M1 subtype is oxoteremorin. Oxoteremorin is the agonist for M1 subtype. But antagonists for M1 subtypes are pyranzepine and telanzepine. Pyranzepine and telanzepine. These two antagonists, they can bind to the M1 subtype on the stomach, and by binding on the stomach, they can block the M1 receptor and they can decrease releasing or secretion of HCl, and they can decrease the acidity in the stomach. The second subtype is M2 receptor. The agonist for M2 receptor is methacholine. Methacholine. This is the agonist for M2 receptor. It means that it can bind to M2 receptor on the heart. By binding to M2 receptor, it can show inhibitory effect. It means that it can decrease heart rate, it can decrease the force of contraction. But the antagonist for M2 receptor is tripitramine. Tripitramine is the antagonist for M2 receptor. It can block the M2 subtype and it can prevent decreasing the heart rate and force of contraction. And last is M3 subtype. The agonist for M3 subtype is beta-nicole, and antagonist for M3 subtype are dariphenacin, and also another is toltrodine. These are the agonists and antagonists, which we will study about these drugs in cholinergic and anticholinergic drugs. This was about cholinergic receptors. Today we talked about cholinergic fibers, different subtype of cholinergic receptor, different type of cholinergic receptor, different subtype of nicotinic receptor, different subtype of muscarinic receptor. And also we talked about the agonistic and antagonistic drugs for these different subtype of nicotinic receptor and muscarinic receptors.